Welcome. In this session, firstly, I'm going to discuss about applications and future challenges of HGP. Then later, I'll also start discussing about DNA fingerprinting. So let's begin this session with applications and future challenges of HGP. So in what way the HGP is useful? So already we have come to know the locations of various diseases like Alzheimer's disease, Huntington's chorea, and then hemolytic anemia, cystic fibrosis. There are so many diseases for which we don't have a permanent treatment. Okay, so here we located genes for various diseases on chromosomes. And now, with the help of gene therapies, with the help of gene therapy, so gene therapy means where we can replace a non-functional gene with a functional gene. So whatever the disorders that are inherited through these genes, they don't have permanent treatment. Okay, so to do that we need to replace that non-functional gene with the functional gene, then that will be a permanent solution. So with the help of these gene therapies, okay, so the certain diseases can be treated completely or permanently. So certain diseases can be treated permanently, of course. So that we need to do it at embryonic level, so not after the birth. There is nothing like the tablets or injections that can cure those genetic disorders, and to cure those genetic disorders permanently, then we need to do at embryonic level. Okay, right. And then the person will have a permanent cure for that particular genetic disorder. So that is what the future of treating the genetic disorders. Of course, we have studied so many like color blindness, hemophilia. At present, there are no medicines or treatment, permanent treatment for curing those genetic disorders. So it is possible only through the gene therapies. Then the HGP can also revolutionize modern medicine practices. So it can revolutionize modern medicine practices. So it can revolutionize, okay, modern, okay, medicine practices, and make drug action more effective, and make drug action more, okay, effective. So that is another the application of HGP. And then, okay, so even knowing about the DNA sequences of organism can actually lead us to understand their natural capabilities, and actually help us to solve challenges in, of course, health care, okay, and even in energy remediation, energy production, in many branches. Okay, the HGP has its applications, and help in studying all genes at a time. So now we have all genes at our hand, okay, and we can study all genes at a time. Like for example, all the transcripts in a particular tissue. So when we see here, a liver tissue, or an intestinal tissue. So in this case, a liver tissue, of course the DNA will be same in all the cells of a particular individual, but in the liver tissue there will be certain genes which will be active, and in the intestinal tissue or in the skin cells, there will be other genes that will be active. Okay, and other some genes will be active. Okay, say suppose we have genes like A, B, C, D, E, F. In this case of the liver, say thus B and D are active, and in case of say a muscle tissue, in this also it will have same genes, but in that case, say this A, C and E genes are active. So that's what, and if they are active, they are going to produce primary transcripts by the process of transcription. Okay, so that transcripts in a particular tissue or organ or even tumor, right, we can study them, and then okay, we can also see that how tens and thousands of genes and even proteins that all work together in an interconnected okay manner, right. So that's some applications and feature challenges of HD. Next we'll see about DNA fingerprinting or DNA profiling. So DNA fingerprinting or DNA profiling. So here what we mean by this DNA fingerprinting. So this is identifying individuals at genetic level. So identifying individuals at genetic level. So that's what we call DNA fingerprinting. Okay, and of course the DNA of a person is as unique as our fingerprints. That is the reason why we have used the word fingerprinting. So DNA fingerprinting. And now we know that when we studied human genome project, out of the all DNA, 99.9% of base sequences will be same in all human beings. 99.9% of base sequences are same in all humans, are same in all humans. So what makes the difference is just that point 0.1 okay base sequences are different. So point 0.1 base sequences are different. Okay, so now if we take the total as 3 into 10 to the power of 9 base pairs of sequences, in that the point 0.1 will be okay, the point 0.1 of this 3 into 10 to the power of 9 will be okay, so 3 into 10 to the power of 6 base pair. So this is okay 0.1% of, right, so DNA, 0.1% of DNA sequences, and this is enough to identify two individual persons. Okay, so of course no two individual persons will have same sequences here, right. And now trying to identify the differences in these sequences, 3 into 10 to the power of 6 base pairs, so we can come to know whether those two persons are of same or different, like how we use it in forensic science. And now this DNA fingerprinting actually involves identifying the differences. Okay, so how we are going to identify two different individuals. So this DNA fingerprinting will involve identifying a different, the differences in some sequences of DNA. So that is called repetitive DNA. And this repetitive DNA doesn't code for any protein. So it doesn't code for any protein. So the repetitive DNA, which means there are some particular sequences of bases which will be repeated again and again on different chromosomes. On chromosome 1, it may repeat some 5 times, okay, and chromosome 6, it may repeat, with 10 times, and so on and so on. Of course, the number also changes between paternal and maternal chromosomes. Okay, so the repetitive DNA, we know that it is not going to code for any protein, and right. So this repetitive DNA will be actually a small stretch of DNA that is repeated, in fact, many times, and these repetitive DNA can be separated from the bulk genomic DNA when we do density gradient centrifugation. So when we do density gradient centrifugation, so what happens is you will get some bands. So you may get some bands like this, okay, and this is a band. So some bands you will be getting like this. So this is the density gradient centrifugation. Okay, so when we do the density gradient centrifugation, what happens in this case, the bulk for the DNA, which will form a major peak. Okay, so this is the bulk genomic DNA, right? And the repetitive sequences will form, okay, small bands. You can see these small bands are actually repetitive sequences of various, the each sequence maybe will be different. So these are bands of repetitive DNA, and these bands are actually called satellite DNA, So I say indirectly that satellite DNA will be having repetitive DNA. So it has repetitive DNA, right? So whatever that repetitive DNA, when we do density gradient centrifugation, it is going to form some small peaks, and other than the repetitive DNA, it forms that bulk, okay, genomic DNA. It forms the bigger band. The remaining all other small bands referred to as satellite DNA. So that consists of repetitive DNA. Now these satellite DNA can also be classified. The satellite DNA is classified on a particular criteria, like the base composition, classified on one base composition. So that may be AT rich or GC rich, means whether we find more adenine thymine base pairs or we find more guanine cytosine base pairs. So basing on that also you can classify this satellite DNA. So base composition, and then the length of segment, length of segment, length of segment, and another one, the number of repetitive units, the number of repetitive units. So which means how many times it is repeated. So the number of repetitive units, and basing on this criteria, we can classify the satellite DNA. Okay, so right, so based on this satellite DNA is classified into micro satellites. Okay, so there will be micro satellites, means it all depends on the number of base pairs present in it, and then like mini satellites. Okay, so the microsatellites usually will have some 2 to 6 base pairs that are repeated many times. So when we see like CGC, okay, TA, now this is repeated again, CGCCTA, you can see the number of base pairs are, okay, 6, 1 2 3 4 5 6, and of course that will range from 2 to 6. In another case you can see uh, CA. Now this is repeated again, CA, and so many times it will be repeated. Okay, so the microsatellites will usually have some 2 to 6 base pair length, and they will be repeated many times. So in this case, the base pair length is 6, and in this case it is just 2. Okay, and in some cases it can be 3, in some cases it can be 4 or 5. Okay, and to maximum of 6. So based on certain criteria, we classified the satellite DNA, okay, into microsatellites and mini satellites. And these sequences show a high degree of polymorphism, means, okay, in some person you may see the same repeated many times on particular chromosome and another one, okay, but the same number may not be seen in another individual, and there may be some mutations in this sequence. Okay, so any mutations that take place in these sequences do not affect the individual's phenotype, because I told you that these repetitive sequences, they do not code for any protein, so hence there will be no effect on the phenotype even though there are mutations. So of course mutations will go on taking place, okay, in these repetitive units. Okay, of course if the mutations take place in the somatic cells, they cannot be passed on to the next generation. If the mutations take place in the sperm cells or in the egg cells in these repetitive units, then they can be passed on to their offspring. Okay, all right, so here these sequences, which actually form the large portion of human genome, and they are going to, I mean, they actually show high degree of polymorphism. Okay, and that is the basis for the DNA fingerprinting. So here the word polymorphism we need to use when we are comparing different individuals. Okay, so then we use the word polymorphism. Okay, so the repetitive sequences, whatever that mini satellites and micro satellites, they form large portion of human genome. So these micro satellites, mini satellites, in fact directly I can say that the repetitive portion of the DNA that forms the major portion of the human genome. They form major portion of human genome. We know that less than 2% of bases will be coding for, or the genes will be coding for proteins. The remaining 98% will not be coding for any proteins here, and they usually consist of a lot of repetitive sequences, and mutations in them are not going to impact the phenotype of individual because they're not coding for any protein. So microsatellites, mini satellites, and whatever, etc., the repetitive DNA, they form major portion of human genome. And these sequences, that is mini, micro, whatever the repetitive sequences, they show high degree of polymorphism. They show high degree of polymorphism. Say suppose, okay, in an individual, so this is a chromosome number 7, this is a paternal chromosome, and we have another one, maternal chromosome 7, maternal chromosome. So paternal chromosome and maternal chromosome. And say suppose there is a particular sequence like CGATC, okay, and this is repeated. So 1, 2, 3, 4, 5 times on paternal chromosome, and on maternal chromosome it is repeated, okay. So 1, 2, 3, 4, 5, 6, 7, 8 times. Okay, it can be it all depends upon, okay, just I have given an example. In paternal chromosome it can be more, and in maternal it can be less, whatever, okay, or it can repeat only, okay, 3 times in this case, and in case of paternal chromosome it has repeated some 40 times. It all depends on, right? So here they show high degree of polymorphism, so both on the length and the sequence of bases. So that's what I say the high degree of polymorphism means, okay, both the length and number of repeats. So both the length and number of repeats. So this is actually the one that forms the basis for the DNA fingerprinting. Okay, so on chromosome 7, this is what you have seen. If you take chromosome 16, in that case of chromosome 16, there will be another sequence. Okay, so TAGC, TAG, some sequence you have seen, and this is repeated, okay, some 10 times on this. Okay, I'll say 1, 2, 3, 5, 6, 6 times it is on chromosome 16 of paternal chromosome, and maternal it is repeated some 4 times. It all, so that's what I say the high degree of polymorphism. So this is individual A, and say this is individual B. So you can see there is the length in this case, and the sequence is different, and the length is different, and the number of repeats can also be different. Okay, so that can be and even if you see on the same individual also, if you are considering on individual A itself, on 7th chromosome this is the condition, this is the repeat of this sequence, and on 16th chromosome, say suppose this is the repeat number of repeats. So the length and number of repeats, that's what the high degree of polymorphism. So this will not be, if you compare this 16th chromosome of another individual, the number may vary. Okay, so means the number of times that is the copy number may vary, and even on this case it can be same in some cases, but at one or other point when you compare with the all, okay, 23 pairs, so there will be at least one or other chromosomes that show the differences in this length and number of repeats, right? So now we'll also try to understand what actually is this polymorphism. Okay, as we have came to that these sequences will show the high degree of polymorphism, so let's understand what actually is this polymorphism. Okay, so here the polymorphism is simply that variation at genetic level, variation at genetic level, and these variations at the genetic level arise due to mutations. Okay, and these arise due to mutations. So there will be frequent mutations in those repetitive sequences, but it won't affect the individual. As I told you, that they are not going to code for any protein. Or in other words, I can say allelic sequence variations. So allelic sequence variations is also called the DNA polymorphism. So you can say it is a DNA polymorphism. Okay, so allelic sequence variations. Now, and in humans, if one variant, so one variant which means allele, we know the variants of genes, what we call alleles. Okay, so if one variant at a particular locus, okay, in the human beings or in human population, if we observe their frequency greater than okay 0.01, then we can call it DNA polymorphism. So the allelic sequence variations, right, if okay more than, so if okay more than one variant, if more than one variant, or the variant which mean allele here, okay, particular at a particular locus, at a particular locus in human population, at a particular locus in human population with frequency higher than 0.01, then you can call it DNA polymorphism, right? Say suppose if I'm going to take some sequence CCGAT, okay, GT is the sequence, okay, and in another person, say there is only one nucleotide chain CCG, and here it is replaced with something, okay, ATGT. So you can see in this case, okay, this is also a DNA polymorphism, and as there is only one nucleotide difference, I can call it okay, SNP, single nucleotide polymorph. So single nucleotide polymorph. Okay, so say this is individual A, is this is individual B. There will be frequent mutations that take place in these repetitive DNA, right? And you can see this is a mutation, so there is a single nucleotide mutation, you can see. So if you find in some different individuals, say some 10 individuals, okay, and at the same point you may see the difference in the nitrogen bases, the nitrogen one nitrogen base difference, okay, so then you can call that as a single nucleotide polymorph. Of course the DNA polymorphisms can be of different types, okay, so DNA polymorphisms can be of different types, like you can take RFLP, so this stands for okay, restriction fragment length polymorph. So it's called restriction fragment length polymorph. Or you can also take VNTR, so that stands for variable number tandem repeat. So variable number tandem repeat, okay, so that also we can consider in this case. But like this, there are different types of polymorphisms. In this case I have given an example of single nucleotide polymorph. This is an example for SNPs. Even this SNP also you can keep here. So that's already, single nucleotide polymorph, are all examples of that DNA polymorphisms, right? So we understand that the length of the sequence and the number of times it is repeated. So out of the 46 chromosomes, okay, that will be having 23 paternal and 23 maternal chromosomes. Okay, so the paternal chromosome may have this sequence repeated some 5 times and maternal repeated, okay, 10 times or only 3 times or 4 times. It all depends on, like this same sequence may be present on another chromosome also, and there the number may be different. And like this, when we compare the length and the length of that one and the number of times it is repeated between two individuals, we can easily, of course, every cell of our body will be showing the same amount of DNA polymorphism. It is not like the skin cell shows a different different polymorphism, or the heart cell or some other cell will show different. So every cell will be at the, okay, it shows same DNA polymorphism, and it will help in comparing two different individuals. Okay, and of course, if the individuals, the DNA polymorphism, so it will be in millions and millions of people. When you compare, then there may be, what you say, that's a very rare case that we'll discuss it later. Okay, but at instance when we compare two individuals, their DNA polymorphism, you can see the difference in the variations in the length and number of repeats. So that is the reason we use word polymorphism. Means polymorphism, whenever we use the word, it is okay when we consider the human population, right? uh, Anyway, so with this I will end today's session, and in the next session we will again come up with this DNA polymorphism with small information, and then we'll discuss the actual procedure of DNA fingerprinting. So how it will be used to identify two different individuals.